actin really only functions when it's in the polymer. And in the cilia that exist in the hair cells of in our ear, they form this really important structure where, has anyone learned about hair cells in the inner ear before? Oh, it's so cool. So in the inner ear, one of the ways that hearing happens, 352, words are hard. One of the ways hearing happens, we have this membrane that has all these hair cells that have this cone-like structure, right? And on top of them is a fluid-filled canal. And so when we hear sound, the sound waves vibrate that fluid, and it causes the membrane to move. And the membrane will touch the hair cells, and that transmits that signal, mechanotransduction, I think, that signal to neurons, and that can basically transduce that mechanical stimulation into perception of sound in our brain. So that's why when people are like, turn the music down, or like when I was a kid, my mom was like, don't stand right next to the speaker when you go to that concert. And I was like, whatever, Mom. And I'd stand right next to the speaker anyways. Like, I was like, 2 minutes to the speaker. Lame, I should have listened. Oh my gosh. Because what happens is when the sound waves vibrate, the fluid too much, it'll hit down really dramatically on those hair cells, and it can cause them to basically smush, and it can be irreparable. So the hair cells will never see nothing, right? Which is probably why I'm so loud now, and I'm constantly like, what? What? Yeah. It's so funny. Okay, no. My Jack is very like safety first. My husband, anytime we go to a concert, he's like, I brought these earplugs for you. I'm like, ugh. Okay, Dad. But I love that he's so safety first. I say that. No, that was the first children's. Okay, so here's the review to go through, and now 20 more minutes. I feel like I'm jumping back. Wait. I had a 2-hour meeting right before this. So boring. Meetings. Okay. Then also, like, it ended like 5 minutes before this class started. Really had to go to the bathroom, and so now I really have to go to the bathroom, which is like down here. I feel like my kid is doing like the bathroom. How many TMI moments can I have in one class? Okay, so now we're going to talk about how protein structure determines function. It really gets back to kind of these foundational principles that I think I've drilled into you already, right? Structure, function. We're going to talk about, protein biochemistry dictates structure, so that's going to dictate function. So we'll talk about that. And then we'll talk a little bit about, really at the end of the day, proteins they're always interacting with something else. It is almost always their job. They're binding to some other molecule, and those interactions are always specific, and that is what indicates their biological role. So we're going to talk about that, give you 1 or 2 examples. And then we're going to talk about how cells control protein function. There are a lot of different ways, so I'm going to go into more detail on what I just introduced, right? Covalent modification, proteolysis, we'll talk about allostery, non-allosteric binding partners, how they can block critical binding sites, interfere with polymer formation, or interfere with the ability of these complexes called scaffolds to interact with one another. So first, TAC A. So the key concepts, right? So protein's biochemistry dictates its functional activities. And that's because protein biochemistry determines its structure, right? If it has a stretch of hydrophobic proteins, it's probably going to get shoved into the membrane, right? And that's going to influence its function. Or where the hydrophobic moieties exist are going to influence how that protein folds. It's going to influence its structure that determines its function. And so regulation of protein structure and function is one of the most fundamental ways that a cell can influence the activities of proteins, right? That dictates the activities of the cell itself. And one of the examples that I'm going to come back to time and again, not just in this lecture, but in the course of this class, is sarcomere. Right? So the sarcomere is a tactile unit of striated muscle cells, right? So skeletal muscle and heart muscle. And the way that it works, for the level that you need to understand it works at this point, right? We have the kind of black and white are a bunch of myosins, right? And myosins, bless you. Myosins have these heads that bind directly to actin. So actin is shown here in green. And myosin is known as a motor protein because it can motor along that actin. So the myosin heads can actually walk along the actin. Totally fantastic. And so what happens in a sarcomere is these myosins are either prevented from associating with actin, they can't bind the actin and they can't walk along it, or they're triggered to walk along actin, right? That's how a sarcomere functions. And so right now I know this is a relaxed muscle because myosin, it's fully stretched out, and when it is triggered to contract, what happens is these myosins on either side are going to walk along the actin, and that force of them walking along it is going to move this whole thing to the midline, right? So as it's walking, it's essentially kind of pushing it backwards, and that's happening in this direction too, right? These are walking this way, pushing this side in. These are walking this way, pushing this side in. So that happens, right? And so we're technical terminology, that happens. And I'm going to talk about how that's regulated by a muscle cell. How do we prevent myosin from associating with actin? How do we trigger myosin to associate with actin and walk along it, right? We're going to talk about that. Okay, so protein structure determines function. And in the book they have these images where the black is supposed to represent this kind of long peptide backbone. And it's just featuring the side chains of, what is it, 6 amino acids. And what they're trying to convey in this image is that you can have functional amino acids, right? And these are the amino acids that are responsible for, if it's a kinase, phosphorylating the target protein. Or if it's a protease, it's responsible for binding and cleaving its target protein. Those would be the functional amino acids that are performing the function that that protein is known for, right? So a lot of times they're widely distributed on a peptide. If I was to draw a peptide out from N-terminal to C-terminal, you'd see a functional amino acid at position like 40 and 70, and 100, 110, like they're right, not necessarily right next to each other in the primary sequence, right? But when that protein folds, a lot of times it can bring all those amino acids right next to each other into a central cavity. So you see that happen all the time. And that's really important for protein function, that appropriate bringing together the functional amino acids, because they can influence each other's reactivity. And so in this image, it's showing, again, you have all these other amino acids present, but it's only highlighting the serine, histidine, and aspartic acid. And so in this image, aspartic acid. So this is a famous catalytic triad, these 3 amino acids. And it's famous because what happens when these are positioned just so, the aspartic acid essentially steals a hydrogen from the histidine. And that causes it to essentially react with the serine. So serine usually has a hydroxyl, right? A hydroxyl group that's pretty unreactive. But when histidine rips the hydrogen away, it creates a reactive serine that's negatively charged. That sucker is what is responsible for cleaving peptide bonds. This is the catalytic triad that you see in many serine proteases. They're named serine proteases because you get this reactive serine that attach peptide bonds. And so the take home of this image is if these amino acids weren't positioned just so, because of the way that protein folded, you lose that functionality, right? How these amino acids are positioned determines the activity of this protein. So, right, what would happen if this serine was positioned over here? So, do you think it'd be reactive? You'd lose proteolytic activity, right? Because that serine, the whole reason that it's reactive is because it's positioned just so next to the histidine, which is positioned just so next to that aspartic acid. If you change the position of any three of those, it destroys this interaction. So that's really the take-home. How amino acids are positioned determines protein activity. That makes sense? Okay. So this idea that all proteins bind to something, right? That's if the protein just didn't find anything, what's it doing? Right? So even like proteins that are structural proteins, they're forming polymers. They're binding to each other. Like I'm thinking about keratin, right? In our stratified squamous epithelial layer in the skin. No, you're not allowed to forget that. It's a structural protein, and its role is the keratin's an intermediate filament. So you have many subunits that intertwine with one another to form this structural purpose within those. Really have this very strong structure that doesn't, strong structure within this simple or stratified squamous cells, binding to one another and binding to the sides of the cells to lend structural integrity to those cells. Another example, right, that's written on the slide is antibodies, right? Antibodies are proteins that they don't just bind to viruses and bacteria. They have other roles as well. They'll bind also to sometimes infected cells, right? So antibodies are molecules that function within our immune system to thwart infection, right? So they bind to pathogens. They bind to things that need to be eradicated by the body. That's their biological role. Wnt proteins, I talked about Wnt proteins. Their biological role is to bind a specific cell surface receptor to trigger signal transduction that, depending on the cell and the developmental context, might regulate intestinal stem cell proliferation, right? So all proteins interact with some other molecule, and that interaction might be really strong or might be really weak, but it is intended to be very specific, right? So let's go with the idea of the Wnt ligand, right? It's a paracrine signaling molecule secreted by cells. It doesn't just bind to any old receptor. It's a very specific receptor that it binds to. So binding by molecules is always very specific. It's like me at a park with a bunch of kids. The hugs that I give out are very specific to my kids, and that tells my function. I'm their mom. Any other kid's mom, because I don't know what to do with them. I still think they're kind of gross and stuff. Another TMI moment. It's true. Those are other, anyway, okay. Well, I never thought I was like mom material because I didn't think I had like I'm doing it again. I just, it's a 2-hour meeting before this, man. It's hard. Stay on track. But I was the person who'd be in a group of people and they'd see a baby and everyone would be like, oh baby, oh my gosh, isn't it cute? It smells bad. And like, do you want to hold the baby? Cool on that. And then, then I met my person, and yeah, maybe I could do this whole marriage thing. He's pretty awesome. And then I was like, maybe I could do this whole making a human thing. Like, that seems kind of cool to like combine our DNA and see what happens. But the whole time I was pregnant, I was like, yeah, this is cool. But I did, I go to these mom groups and like, oh, are you connecting with your baby? I'm like, no, I'm thinking about developmentally what's happening. Like, that's totally fascinating to me. But then, like, the second he was born, I was like, oh my gosh, he's amazing. Like, look at him breathing. He's breathing better than any child has ever breathed in the history of children. Like, you just like, oh, it's so amazing. And so that I very specifically bind to my children. I still don't get other children. That's okay. I'm fine with admitting that. Okay. Cellular control of protein function. So proteins, right? They bind to these other molecules, but a lot of times you want that interaction to be really specific. We don't always want Wnt binding to its receptor. We don't always want muscle cells contracting, right? So how do we regulate those interactions between proteins? How do you regulate their function? There's all these different cool ways that a cell can do it. And I've given you some of the tools to think about this already. First, covalent modification, right? This idea that you can have an enzyme that adds a chemical group to a protein and it totally changes its function. And so covalent modification, the most basic definition is enzymatic addition of some chemical subgroup. And it's added via covalent bond, right? And so things like phosphorylation, glycosylation, lipid modifications are all covalent modifications. And again, there are a lot of other covalent modifications. These are the three that I'm going to talk about right now. Yes, right now. Okay, so first and foremost, phosphorylation. That's what we're seeing in this image right here. So we have this globular protein with a serine side chain, a protein kinase, right? That is the name of the enzyme that phosphorylate proteins. It will add a phosphate group via a covalent bond to that amino acid. And then if you want to remove that phosphate group, that has to be performed by another enzyme called a phosphatase. And so in some instances, protein phosphorylation can turn a protein on, right? It can, in some cases, right, kinases will phosphorylate a kinase to activate the phosphorylating activity of that kinase. Or maybe you're phosphorylating, a transcription factor, and that leads to its ability to bind DNA, right? So that would be turning on the function, allowing the protein to perform the function that it's designed to perform. And in that case, if you dephosphorylated it, you'd be turning the protein off. It doesn't always go in that direction, so sometimes phosphorylating a protein actually turns it off. A phosphatase to turn it on. So it's just to say protein phosphorylation and dephosphorylation, sometimes it might turn a protein on, sometimes it might turn it off. Right? And we'll talk about that, specific examples of that as the class proceeds. Yeah, Alfredo?
When it comes to function, you were talking before, it was like some proteins like fire have strong bonds and others
weak.
Bonds.
Yeah.
Does either weak bonds or strong bonds have anything to do with function?
Yeah, that's a great question. So protein-protein interactions are usually going to be transient, right? Nothing is forever. And so if you have the way that a strong bond is actually determined is the rate of dissociation. So how how long do the proteins interact with one another once that association forms, right? How long does it take for it to dissociate? So a lot of times if you have a weak interaction, the function is going to, probably not last as long because it's going to dissociate faster. That makes sense. Good, we can go into much more detail on office hours, talk about pKa's, it'll get wild. Okay. So eukaryotic cell contains a whole bunch of different protein kinases, a ton of them. We'll learn about some of them when we get into signaling. Signal transduction pathways heavily rely on very specific kinases that have, either a specific target protein or, kind of a specific subset of target proteins. So there isn't like one kinase that phosphorylates everything. They're really specific. Is kind of the take home. And then there are also phosphatases. Glycosylation, addition of a carbohydrate group, right? Big old sugar. In-linked glycosylation. There's also O-linked. We'll talk about that a little bit more when we get to the Golgi. In-linked always happens in the ER. And it occurs once a peptide moves into the ER lumen. What's this protein called right here? Blue one. Protein translator, yes. Sing it to the world, Jomari. Sing it to the world. No whispering here. Be great. So protein translocation. It is just water. This is just how I am. I swear there's some people who are like, she made that vodka joke or you said. I had a high school professor who was in class. So the peptide enters the ER lumen, and there in the ER membrane is this enzyme, right? That's this big old mouthful to say, oligosaccharyl transferase. It's a big old mouthful to say, however, it still tells you what it's doing, right? So ACE, it's an enzyme, transfers, cool, it's transferring something. Oligosaccharyl, it's transferring an oligosaccharide, right? Oh yeah, it would have been nicer if they said oligosaccharide transferase, but they didn't. Oligosaccharyl transferase. It transfers an oligosaccharide to an asparagine residue. What's the one-letter code for asparagine? N. Hence, N-linked glycosylation. It involves the enzymatic addition of an oligosaccharide in the ER by oligosaccharyl transferase. Okay. So, you guys have a quiz again. It was supposed to be just on lecture 10, but I barely got into lecture 10, so I'm going to add questions on. It's going to be like a lecture 9, maybe beginning of lecture 10. But it's